So, in the dark reaction process, in the process which is called the Kelvin cycle, which takes place inside the stroma of the chloroplast, the carbon dioxide is fixed into the by the Rubisco enzyme into the ribulose bisphosphate molecule, forming finally the first stable component as 3-phosphoglycerate. This one, 3-phosphoglycerate, the first stable component of the carbon dioxide fixation, the Kelvin cycle process. And this is the reason this particular formation of this three carbon structure, the phosphoglycerate acid or 3-phosphoglycerate, is actually the one which leads to naming this entire process as the C3 cycle. It's very common since it's actually the one which is present in most of the organism that we see around, and it's actually the method by which the carbon dioxide fixation it actually takes place. But it has been noticed that under certain condition, some of the plant, even to some extent, there is a wide variety of them that are there, which actually does not follow this method but has switched to a complete different system. This the main reason behind that being a process called photorespiration. Now, for those like plant, what actually happen is they lose the CO2 and lose a large amount of water actually, and for that they have designed this particular method of a more efficient way of fixing of the carbon dioxide. And this happens by due to this particular structure within their cell. See this arrangement that we are seeing here, this is actually the bundle sheath, means the vessels, the all the vessels that are there inside the um, leaf. Right surrounding it we are going to find there are something called this bundle sheaths are there, these cells, and surrounding it are the mesophyll cells. If you remember, it is a mesophyll layer which is responsible for fixation, for the process of photosynthesis mostly. But just before this mesophyll cell in the bundle sheath, there will be bundle sheath surrounding it, and this structure is what goes by the name of Kranz anatomy. So this Kranz anatomy that we are seeing here is actually the one which helps in the more efficient fixation of the carbon dioxide in those particular set of plants. And this process is actually considered is mostly seen in the tropical climate, because the process which triggered that photorespiration where excessive amount of water is lost, inefficiency even of the C3 pathway, even we can say that photorespiration process is what is triggered by high temperature which is like around 95 degree Fahrenheit and so in this hot climate, in hot tropical climate, this particular thing is quite prominent, means this particular anatomy within the leaf is quite prominent. So the process, what actually happened, this process it goes by the name of the C4 pathway instead of C3, because the process actually involves fixation of carbon dioxide not by ribulose bisphosphate, not into the ribulose bisphosphate, but rather into a molecule called phosphoenol pyruvate here. This one, PEP, is called phosphoenol pyruvate. It's a three carbon structure, and this PEP actually is with the one which the CO2 gets absorbed or bind into, and the one responsible for doing that process is this particular enzyme called the PEP carboxylase, or the full name will be phosphoenol pyruvate carboxylase. So what do we get out of that? We get this particular chemical. It's a 3 carbon PEP with which a carbon dioxide is adding, and now we are getting oxaloacetic acid or oxaloacetate as the first stable component, and it is a 4 carbon structure. That's why this process it goes by the name of C4 pathway. Now remember, this step is taking place inside the mesophyll, and is actually a method which deviates the process of Kelvin cycle that we normally know. So once this process takes place, so this mesophyll cell, we find the PEP is getting converted into oxaloacetic acid due to addition of the carbon dioxide with it. Now this oxaloacetic acid will get converted into malic acid or malate, this one, right? And once this malate is formed, malate will actually leave the mesophyll cell and will enter into the bundle sheath cell. Okay, means it's actually transported from the, it simply gets diffused out from the mesophyll straight into the bundle sheath. Once it enters the bundle sheath, it actually release this carbon dioxide. Okay, and the pyru means malate, it releasing carbon dioxide gets converted into a component called the pyruvic acid or the pyruvate. Now this pyruvate will be converted into phosphoenol pyruvate, uh, and at that time an ATP will be utilized in this particular process. Now, in the bundle sheath, we are going to find that the CO2 that is coming out, means the CO2 that is being released by the malic acid, that CO2 is going to get fixed further by the process of Calvin cycle again. Means in the bundle sheath, we are going to see that the Calvin cycle is taking place. And this Kelvin cycle process is actually again the C3 process that we know, that one that I've shown you previously, where let me show you again. Here the CO2 gets fixed into RUBP, means ribulose bisphosphate, finally becoming phosphoglyceric acid, finally becoming bisphosphoglycerate, then into glyceraldehyde 3 phosphate, which is like 6 molecules of them are going to form, and then finally from there we are going to find that it is getting converted into, means one of them is going and entering into the process of the reverse glycolysis process, in which they actually get transformed into the sugar, desired sugar molecule, and right there they become desired sugar molecule. The rest 5, they enter into the process of regeneration of the ribulose bisphosphate. That way the cycle actually it takes place. But in this case, we are going to find that the same thing is going to happen right here in the bundle sheath, and then finally the sugar is formed, which is transported into the vascular tissue. And vascular tissue, as you know, is like the phloem cells and all, so they will be responsible for transporting it to the storage organ. Now this entire process, this entire step, it actually is considered more effective because of this addition of CO2 into the PEP carboxylase, like by the PEP carboxylase into oxalic acid, is actually considered as comparatively more efficient, and this process is considered a bit more efficient in fixing carbon dioxide than that of the uh, process of this C3 pathway normally. So we are seeing here two-time fixation of carbon dioxide, isn't it? First time taking place here in this particular step with the PEP. The second time taking place right here with the RUBP actually, because it is the Kelvin cycle process again. So it is RUBP which is with which it's getting fixed into, right? So there are two steps of fixation of the CO2 is taking place. It is it is not as energy efficient as we can say the Kelvin cycle itself is, but this the efficiency of the fixation of carbon dioxide is comparatively higher. Okay, and even there is no loss of anything due to photorespiration in this particular process also. So that is the process of C4 pathway, and it is the plant mainly they enter into this particular process where the two step fixation of the CO2 will take place there, with the C4 molecule forming in between, is actually something that happens in the tropical climate, and the maize, the corn plant and all, tropical grass and all these things, whereas the C3 is like the like normal region everywhere, all the other plant and everything we can find them. There is actually another process that exists which is called the Carnosian acid metabolism, and in this there is a different way this actual thing happens. Means the fixation takes place slight some a little different manner. We will be discussing about that, the C2, the proto respiration process and everything in a later video.